Hello, in this video we're going to be talking about DNA replication. So let's not waste any of your time. Let's get right to it.
It's hard to breathe, but that's alright. Hush.
Now we're talking about DNA replication. Remember, this is information that was gleaned from experiments from the scientists, 1800s, 1900s. But it's the idea of when they were looking at DNA replication, it could be happening in one or two or three types. The semi-conservative, which we already know is where we landed, where there is a mixture of the old and new in the double helix, one old, one new. The conservative, which was the two parents paired back together and the two new ones paired back together, or dispersive, or the intermediate, which was what was called intermediate on the previous video, where there's actually a mixture of the DNA, and we know enough about that process to know that although that may happen during meiosis, where there's a little bit of a mixture on the chromosomes, it doesn't happen during replication. And so we know that semi-conservative is the pattern used. All right, so yes, as we just mentioned, that replication process, both DNA strands are copied, and then the parent DNA and the new copy DNA paired together to go back to keep that semi-conservative. If nothing else, it always keeps the DNA structure young, for lack of a better term, because every time I copied it, if I always put the newly strand copy with the adult, then I keep that set somewhat younger. If it was always old to old, new to new, there'd be some really new ones and then some old ones. So it does its part to keep it as young as possible. All right, the idea of DNA replication, it's a pretty straightforward event. There's a lot of players involved. One of the things I need to say, and it may have already come up, and I didn't notice it in the written text. I don't think so, but it possibly could have. In the text, just for space and time, there was a shortening of the word polymerase, just a POL. So anytime you see DNA POL in the written text, it really stands for DNA polymerase. Just a little not nickname, not abbreviation, not even an abbreviation, but just a shortening of the word polymerase. But okay, so what happens with the DNA? It gets unwound. Okay, and then it gets separated, and we're going to talk about who does that, what does that, as we go through our video. Oh, and just remember it gets read in the 5-3 direction, and I'll show you on a later slide. But it's not the 5-3 on the original set; it'd be the 5-3 direction of the new copied set. Yeah, just remember that it's read 5-3, on the 5' to 3', and DNA polymerase, as you can see, kind of shown by the beige blob on the diagram, is the enzyme that reads and matches up those correct base pairs, the A, T, C, and G. Remember, it's not the sugars or the phosphates that are read; it's simply the nucleotides. So if it sees an A, then it puts a T with it. If it sees a C, it puts a G with it, and vice versa. But DNA polymerase. Now, so DNA replication, it has a fancy name, a replication fork. And the part that we're going to talk about is the far right of that diagram. But we first want to start with, we mentioned on the previous slide that DNA gets unwound. The enzyme that does that is called heliocase. It unwinds it, and then the DNA polymerases attach and begin to read that DNA and matching the correct nucleotide. But the idea of it is this replication fork. It isn't so much that it just unwinds it. Okay, it's read kind of in a loop, because what you want to think about is a DNA polymerase doesn't read the entire strand of DNA from end to end. There are many polymerases that attach, and each of them kind of do their own little section. So if you look at the diagram on the far right portion of it, you can kind of see what this replication fork looks like. The blue line is the flat DNA, two strands. Okay, but then they're separated. Okay, and they kind of go up in a like a loop above and below where the location of the original strand was, and then the polymerases go along there and read it. So the new set kind of is also in a loop, and that leads us back to when it's done and they're laying flat again. Now you can see that the blue and the red are side by side, and that's when they're paired back together. So a lot of it, that semi-conservative nature, it isn't so much that that's what the cell is deciding to do. Hey, keep the two old ones separated and overlap them with the new ones. It's that they're right there, and they are. If you think about it, when they're put back together, okay, they're connected. So when those A's and T's are put there because of their polar nature, okay, there's that hydrogen bond that connects them. So it isn't that so much that the cell decides, hey, let's do a semi-conservative process here. No. When they are matched together, there is that magical bond that just occurs. And so now they're attached to each other. So that's the reason why the old and new are put together. It's not because the cell is choosing to, it's because it's what happened naturally. You wouldn't read it and then have those bonds attach them and then break that and then put the two new ones together and the two old ones together, because they wouldn't. I mean, the old ones would match up, and they would match up, but it's just they're already attached, so it's just left in that manner. So when we're thinking about DNA replication, one of the things is that is done. The polymerase, the start of this needs a little kick start, a little shove to get going. So an object called RNA primers are added to the DNA strand, or as far as reading it. And that just kind of gets the process started. These obviously will include a starting place, starting point. So the DNA polymerase adds, or the primer is added to the strand, and then the polymerases know to start here and begin reading from there. All right, now the terms we want to think about is leading strand and lagging strand, and these are just to differentiate the two strands. So of the original set, when it's unwound, the set or the strand that is the 3' end that is open, that's called the leading strand. And the 5' end is called the lagging strand. Now the leading strand, because it's in the right direction, and remember, if we're reading it, if this is 3 to 5, then that means the upper side is 5 to 3, which is the direction it wants to go. And that direction can be done continuously without any break in the action, because it's going in the direction that the DNA strand wants to go. The opposite, okay, the lagging strand, it's not going in that. You just can't start at the end and get going. It has to start more in the middle and work backwards, if you can visualize that. And that's called the lagging strand, simply because it takes longer, or an idea that lags behind. And because of that, because it does that, it's replicated discontinuously. In other words, it does it in short little sections and then connects those sections, because it starts in the middle and works its way to the end, as opposed to starting at the end, working its way to the other end. Oh, and those little sections are called Ozaki fragments. Simply, that's the name of the person that figured this out. Excuse me. So the rest of these slides, we're going to look at the kind of the same material, just in a different manner, and just emphasize the different parts. But here you've got the DNA strand, the original, the helicase unzips it, and then you've got your replication fork. You got your lagging strand, which is going to be the 5' end, the leading strand, which is going to be have a 3' end on that end. Just remember the discontinuous, continuous, the lagging strand, discontinuous, leading strand, continuous, and the short little primers are put on there to get it going. And then of course one will just go right through, the other will be on a discontinuous manner. Alright, so you can see our leading strand here. When we're synthesizing the leading strand, the steps, the helicase unwinds it, the primers are added to get that little push start, and then continuously working along, the DNA polymerase reads those nucleotides, complementary base pairs, A and T, C and G, and builds that. And of course, this is the one we saw earlier. Looking at that discontinuous, we looked at the leading, and what's coming up now, we're going to look at the lagging strand. So the lagging strand, again, you've got your Ozaki fragments. It's the strand that, when it's opened on the 5' end. And that's the opposite way that it wants to go. So that's what the discontinuous nature is. Okay, again, thinking about the lagging strand, the idea is once it's opened, primase adds the primers, the DNA polymerase comes along and begins to read the DNA and build it, replicate it, and then of course the Ozaki fragments are added, and then a word that we haven't seen yet, the DNA ligase comes along and joins those Ozaki fragments, because they're connected to the strands but they're not connected to each other. So the ligase comes along and completes that process. Alright, and this information just review everything. The leading strand, the primase adds the primers, DNA polymerase comes along and gets that read, and then the RNA primers are replaced by polymerase as it reads it. Lagging strand, kind of the opposite, 5' ends, reading it discontinuously, the Ozaki fragments, and all that's included. Well, that's our video over DNA replication. When we come back next time, we're going to be talking about the telomerases. We'll see you then.